Hi, everybody. Welcome back to our journey in Chapter 6. The first half of the chapter, we just got done taking a look at the growth requirements as far as chemicals and energy and oxygen and nitrogen and other things and physical growth requirements, temperature, pH, osmotic pressure, hydrostatic pressure, those types of things. Now, the second half of Chapter 6, we're going to take a look at actually culturing growing microbes in the lab setting and taking a look at some of the considerations we need to think about for that. So first of all, when we start talking about culturing microbes, we have some new vocabulary we have to understand. So when you take a sample of something, it's called an inoculum. So that's the vocab word that's here in your notes. It's just a sample of a microorganism. It could come from a surface, a non-living surface. It could come from a living surface. It could come from an infected wound. It could come from a mud puddle, whatever. Inoculum is just the sample or the source of where your bacteria is coming from. And then you typically put it on some kind of media. So the media is like your broth or your agar or whatever it is you're going to be growing the bacteria in or on. All right. So usually you'll grow bacteria in test tubes, which are going to contain a broth or a liquid medium, or in a petri dish, which is a solid agar surface that you grow bacteria on the surface. Sometimes you'll see agar in test tubes, either in a slanted version or a deep version that you might stab, like we saw with the oxygen requirements medium. So when you grow bacteria on the surface of agar, like in a petri dish, you can observe what we call colony morphology. Colonies are the little clusters of bacteria that are growing on the petri dish. Morphology is their appearance, the colony morphology, as opposed to cell morphology, like we could determine with staining, like we learned in Chapter 4. So just like your cell characteristics, if it's a bacillus or a coccus or a spirillum, if it's gram-positive or gram-negative or acid-fast or something like that, that's all characteristics you can help identify an organism based on the cellular things. We can also describe bacteria based on their colony morphology. What do they look like when a whole cluster of the bacteria grow together? And what's cool about it is a lot of times you can classify, or bacteria have of similar relation grow in their colonies in a particular way, which is very cool. So you can classify colonies based on their shape, right? Is it a circle? Is it kind of, so for example, these would be circle or round shaped colonies, whereas this guy over here would be considered like rhizoid or filamentous. That one looks like a little bit more filamentous. This one here is probably irregular. It doesn't really fit any of those particular other characteristics. The margin is the actual edge. So this one has a nice smooth or entire margin, where this one is irregular. See, it's kind of bumpy like that. So that would be an irregular, or, you know, all these different examples. And then elevation is if you're to take your petri dish and kind of look at the side, how tall is it? So if you're looking at your side of your petri dish, right, and here's your agar, and if your bacteria is growing a dome like that, we call that convex. Or sometimes it's got like a little bump in the middle, and we would call that umbonate. Or maybe it's just flat. It's just got a little, like a table rock shape. And so that would be a flat or a raised colony. So looking at it from the side would be the elevation. Size, you have really big ones, we have really small ones, and everywhere in between. The texture could be rough or smooth. Sometimes you can see it's shiny, like it's glossy or dull. Sometimes it's pigmented. So here we have a yellow, So here we have a yellow colony over here, kind of a blackish dark blue colony over here. These ones are kind of golden. So color can also be characteristic or classifying of a particular group of bacteria. Some can be opaque or translucent or transparent. So it's all adjectives, right? It's all these descriptive terms to show you, to help to describe what the bacteria look like when they're growing on the surface of a agar. And it also helps you identify if you have any contaminants. So if you're only trying to grow one type of bacteria and they're all kind of nice white, circular, round, smooth margin bacteria, and then you get a big yellow splotch somewhere on your petri dish, that's a good clue that you have a contaminant, and leave that one alone if you're wanting to just focus on your organism of interest. Okay. Speaking of that, of your organisms of interest, there are ways that microbiologists can do, techniques that they can use to try to obtain a pure colony. Because bacteria are everywhere. We can't visibly separate them because they're invisible to the naked eye. So what we can do is we can use petri dishes and agar plates to help us isolate an individual colony. And so this first technique is called a streak plate. You'll get to do this in lab. Is you get your culture on your loop and you spread it really, really strongly in one section of the petri dish. And then you sterilize your loop. And then without going back into your original stock, you take your loop and you just go a few times into that first streak, and then you just bring it out to another section of the petri dish. And then you sterilize your loop again, and then you don't put it back in the original bacteria. We're just dragging what's originally on that prior streak out into a new sterile area of the petri dish, and you sterilize your loop again, and then you do this last streak into the middle kind of area. And your goal is to end up with these individual isolated colonies. And these are called CFUs, which stands for colony forming unit. And what a colony forming unit is, it's usually a single cell, one cell that got drug out from all of your different streaks from your sterile loop, that got drug out and landed there on that part of the agar, and then it goes and it multiplies, right? One cell to two, two to four. So then after a couple days or 24 to 48 hours in your incubator, you're going to get millions of bacteria, and that's what your colony is. And so that colony is the offspring of like one cell that got drug onto that particular area of the agar, and they're all clones of each other. So they are genetically identical. They're not a mix of other bacteria. So it's not going to be a mix of the green and the yellow. We don't want that, because then if we're running experiments, we wouldn't know which organism is, doing our experiment. So having pure colonies is really important. Streak plates is probably the most common mechanism. It's quick, it's easy, it's fast, and it works really well at getting those isolated colonies. Sometimes you need a slightly different way to do that, and so you can do pour plates. Pour plates, you need to do a little bit of a dilution. So you take your original sample, you dilute it a couple times in some broth, and then you mix that broth in with some melted agar, kind of some warm agar that's not solidified yet, and then you pour that into a sterile petri dish. And then you can get your isolated colonies, right? Little isolated colonies, but they're not always growing right on the surface. They could be growing within the agar. And so here we can see some isolated colonies, these big white ones, and we see some little tiny little pinpoint white ones. And so those would be your isolated colonies for those different organisms. Okay, all right. So those are kind of some general ideas on growing bacteria. Another cool thing about growing bacteria is we can adjust, we as the humans and the researchers can adjust the ingredients in this media that we grow the bacteria on, and that allows us to learn some information about our organisms. So I have a wide variety of media pictured here. It can be selective in that it allows only certain bacteria to grow. It can be differential in that the bacteria can grow, but it might grow differently, which tells you something about the organism. Let's see. I have this, make sure I'm hitting all pieces here in the notes. Oh yeah, so the different types of culture media. So defined means you know all of the ingredients in your recipe, down to the last gram of something. Compare that to complex media is you have ingredients like a yeast extract or beef peptone or pancreatic digest of casein. These are all more complex. You don't know all that's in them, but you know there's good, nutrients. It's got all the ingredients. You just don't know the exact quantities of each one. And most the media that we're using in lab is a complex media. It's got all of these kind of digests of yeast and protein and things like that. So then selective media, I'm going to talk about this right here. This is a type of media called mannitol salts. Mannitol salts agar. Sometimes it's called MSA for short. Mannitol salts agar has a high concentration of sodium chloride. So you put in like 15 grams of salt in this liter of agar. And so that's a selective part of it. So we talked about halophiles before, right? So if you were to plate a normal non-halophile organism on mannitol salt's agar, it's not going to grow because all of the salt that we put in the agar plate is going to suck all of the juice. It's going to suck all of the water out of the bacteria, and it's not even going to be able to make any colonies whatsoever. So mannitol salt's agar is selective for halophiles, which means halophiles can grow on it, selective against non-halophiles. They cannot grow on it. Okay, so that's a good example of selective media. Differential media, this is also a differential media because can you tell the difference here between the appearance of the agar? So some agar or media, I should say, because it's not always agar, is of the bacteria that can grow, it turns colors. Either the bacteria itself turns color or the media itself turns colors. And I'll talk about some of these other examples in a little bit. But mannitol salts, it's selective against for halophiles, but it's differential for halophiles that can ferment mannitol. That's the mannitol part. MSA is normally pink. So on this half of the plate, that is what an uninoculated side looks like, or something that doesn't have anything growing on it. It's normally pink because it's got a pH indicator that kind of turns it that pink color. If you're an organism that one is a halophile, so you can grow, two, you can ferment mannitol. What do bacteria ferment sugars into? Acids, that's right. So when it ferments the mannitol into an acid, it releases the acid and turns the phenol red yellow. So that is a differential appearance. So we can see, yes, that organism, that bacteria that's growing on this MSA plate, it is a halophile because it can grow, and it ferments mannitol because it turns the media yellow around it because it's releasing an acid. So that's a good example of selective and differential. This plate over here is one called eosin methylene blue. It's called EMB. It is also it's differential and selective. I don't see the selective part on here. It selects against gram-positive, so they can't grow, but it selects for gram-negatives. And it's differential because look at this cool bacteria grows like this metallic-y, shiny green, where some grow kind of like a purpley, dark purple, and some hardly grow at all. So the ingredients in this media allow us to differentiate between some of the characteristics of the bacteria, whether they can ferment lactose or they cannot ferment lactose. Okay. This particular media, that's a different color, that green's not very bright. This particular media is indicative of an organism being able to use citrate. It's citrate. It's got an enzyme, part of the Krebs cycle. So if it's positive, it turns blue. If it's negative, it stays green. This particular media looks for an enzyme called urease. If it turns this hot pink color, it's positive for urease. If it stays that yellowy color, it's negative for urease. This is another media that we have some differential qualities based on the color of the bacteria that's growing, yellow, orange, or white, and that's going to be indicative of some of its physical or chemical or biochemical characteristics. So this is a fun part. I love these parts of our microbiology labs because we get to grow all these different kinds of bacteria and they turn fun colors and stuff.